Hello students, my name is Minal Chaudhary and today I am going to discuss about one of the biology chapter in class 9, that is cell, the fundamental unit of life. Now the cell is not a new topic for you because you have studied about this in your previous class, that is class 8 also, and if you continue to study biology in future, then you are going to study about the cell in higher classes, for example in class 11 also, and even in graduation. Now, what is it that you are studying the cell topic again and again in all these different classes? This makes us understand that the most important topic in biology is nothing but cell, because this is the most fundamental unit of any living organism. Like if I speak about chemistry, then without understanding atom, you cannot understand anything in chemistry. Similarly, if you want to study biology, you need to understand what is a cell first, then only you can go a step forward in any understanding any other concepts of biology. So, as far as discovery is concerned, in 1665, Robert Hooke, this was the person who has discovered the cell. However, he was observing a cork tissue in the plant bark. Now, this cork tissue is something that you will learn in the next chapter, that is in plant tissues, where you will learn that the cork tissue is actually a kind of dead tissue that you find on the outer part of the tree, that is the bark, which we call as. So, he was actually, without knowing, observing some dead tissues. Now, when you see a dead tissue inside, you won't see anything. You will simply see the outer boundaries. So he could see the outer walls or the boundaries of the cells, which appeared something like this structure, a honeycomb-like structure, and therefore he called it as some compartments because it appeared like small compartments. So these compartments he called in Latin as cellulae, from where the word cells came into picture. Then in 1674, Antony van Leeuwenhoek, he was trying to observe various things by a microscope which was made by him. That was his own made microscope. And for the first time he observed certain living cells and the organisms. Like you have a picture here, microscopic observations by Anton von Leeuwenhoek, some white blood cells, then red blood cells, some yeast cells which is dividing, sperm cells, bacterial cells. These living cells for the first time was observed by Antony van Leeuwenhoek. Now coming to a definition of cell to start with, you call it as the fundamental or basic or the smallest structural and functional unit of life. Now please understand, if you look at the definition, there are two important words: structural unit of life I am calling it as, and functional unit of life, and that too it is the smallest or the basic structure. Now, why is it that you call it as the smallest structural unit of life or smallest functional unit of life? In simple understanding, if we take an organism and we start breaking it down into smaller and smaller pieces, then the last smallest structure which you will observe, that will have an independent existence. Try to understand, independent existence means it can survive independently like that. Such a smallest structure will be a cell. Not that you cannot break down cell, you can break down the cell also, but if you break down the cell further, then that broken cell cannot exist as a complete independent unit. Therefore, a complete smallest structural and functional unit which can function independently is nothing but the cell. Now, these cells make up what are called as tissues. That is your next chapter that you are going to learn. And those tissues make up organs and organ systems. Some discussions that you will have in the future chapters in class 10 also. So this was a diagram from your previous class. I believe you can recall the cells as observed in onion peel are compared to the brick walls. Like your home walls are made up of many such small bricks coming together. Onion peel tissue is also similarly made up of many such small cells which have come together to form tissues. Now, very important topic, cell theory, which speaks everything about what a cell is and what is its importance for a living organism. In 1839, it was German botanist Matthias Jakob Schlingen. He said that every structural part of plant was made up of cells and or the result of the cells. In 1839, this was in 1838, and this in 1839, Theodor Sohn, who was a zoologist, he said that the animals are composed of cells or the products of the cells in their structures. So, comparing both these statements, the first two postulates or tenets of the cell theory was proposed, that is, all living organisms are composed of one or more cells, and the cell is the most basic unit of life. This was the definition of the cell that we said. In 1855, Rudolf Virchow, he said in Latin, omnis cellula e cellula. What does it mean? It means all cells arise only from pre-existing cells. The question came up, where from new cells come? It cannot simply come from nowhere. So a cell, for new cell formation, there has to be already a pre-existing cell. So as in cell theory, we get three tenets, that is, all living organisms are composed of one or more cells. The cell is the basic unit of life, and all cells arise only from pre-existing cells. Now, an organism might have a single cell. It might be made up of a single cell, you call it as a unicellular organism, or it might be a multicellular organism where they have multiple cells. This is not a new concept again. Unicellular organisms are made up of single cells, multicellular they have multiple cells. In unicellular organism, that single cell can carry out all the life processes which are required for its survival. Multicellular organism, it has the cells making up tissues, and those tissues further might make up organs in more complex organisms, where those specialized tissues or the organs will be able to carry out different functions. Unicellular organisms, they divide to form more organisms, whereas in multicellular organisms, they start life as a single cell only, but that single cell can undergo repeated division to form a multicellular structure. Amoeba, paramecium, etc. are examples of unicellular organisms. Plants, animals, we are multicellular organisms. Now, when we talk about shape of cells, one thing is sure, all cells are not of the same size and shape. It can be different. But that difference or variation that we observe in the shape of the cell or the size of the cell depends upon what? Most importantly, it depends upon the functions of the cell, what type of function they carry out. For example, I am coming directly to this particular cell diagram that you see. It is a nerve cell. Now, what is the function of a nerve cell? Is to carry information. So, within the body, within the tissues, it might have to carry information to long distances. That's why you see the cell is elongated. I am coming to this cell, which is a red blood cell. Now, red blood cell is a cell which does not have a nucleus inside. Why is it so? Because its primary function is to carry oxygen by binding to the hemoglobin. So, it needs a lot of space inside. To have more space, the nucleus is absent. So, the cells can be round, spherical or elongated. There are some spindle shaped. These are spindle shaped cells, which means both the ends are tapering. That is, it's pointed. So, cells like muscle cells are spindle shaped. Some are very long. They can be branched also at times. That is, neuron or the nerve cells. And some are irregular shaped. Like, for example, amoeba. It's an irregular shape. It does not have any specific shape. And above that, it can even change its shape as per requirement. Like I was telling just now, these cells can change its shape, the amoeba. They form certain projections from their cell surfaces, which are called as pseudopodia. Pseudo means false and the podia means fake. That pseudopodia helps them to move and to engulf food which would be there in their surroundings, in their vicinity. Now, this is a process, basically how you can see the cell is forming a pseudopodia, and it is in the process of engulfing. Completely it has engulfed and forms a food vacuole. This is a realistic electron microscope picture of a cell. Not only amoeba, some other cells like WBC, white blood cells in human beings, they are also having this particular ability. That's why they can engulf or destroy some harmful things that may enter our body, enter our cell. Size of the cell. The size of the cell is not fixed. It can change. It does not depend upon size of organism. It's not that human cell will be bigger than the cell of an ant. It depends upon the function of the cell to some extent. It may vary from few micrometers to few centimeters. Some of the smallest cells known are bacteria and some similar organisms. And one of the largest single cell can be considered as the ostrich egg. This is a picture where we see a comparison of different cells and certain subcellular structures, that is the structures present inside the cell. We are starting with an atom of 0.1 nanometer and then some molecules like lipids and proteins which are larger than the atom, and then some viruses, flu virus, which is even smaller than some other cell organelles, like mitochondria is a cell organelle, but flu virus is even smaller than the mitochondria. Bacteria, almost of similar shape as mitochondria. Plant cell, animal cell, a human human egg, a frog egg, chicken egg, an ostrich egg. You can see different sizes of the different cells or the subcellular structures. Now, how do you see these cells? Because in the previous slide you have seen these cells are very small, few micrometer. Micrometer means 10 to the power minus 6, that is you put 0. Then 50 then 1 is micrometer. So it's very small. Therefore obviously you cannot see with your naked eyes. Then you will need some specialized instruments. These instruments are called as microscopes. Now to observe these cells you need to color them. There are special chemicals that you use, some dyes that you use to color the cells, and they are called as stain. When you put the stain, the cells get colored differently, the different parts can get different intensity of color, and then you can easily observe them under the microscope. I will show you some pictures later on. Egg of a hen is a single cell which is visible with the naked eye, but not all cells. These are some specialized cells which are large cells. Coming to these microscope types, now this is an extra discussion but very important. Believe me, if you want to study biology in future, then microscopes must be your best friends. So it's important that you know something about microscope, at least something in this class. So there can be different types of microscopes actually. Most common ones are light microscope that we regularly use in our laboratories. Electron microscopes you won't see in your laboratory. If you go to some research laboratory, you go to IITs and all, you will be able to see an electron microscope. There are various other types of microscopes which you will not be able to understand at this level, so I have just kept it as others. Looking at these light microscopes, most common ones that we use, which you will be seeing in your school laboratory also, they are simple light microscope and compound light microscope. A simple light microscope is something like this, where you have only one lens. In a compound light microscope, you will have two lenses here. So there is a greater magnification of the image. This is how a normal compound microscope looks like. This is, however, a schematic picture. You can ask your teacher to show it in your school laboratory. You see here there are two lenses. One is this, which is called as an eyepiece. Three here which are called as objective. Of course we are not using all the three at the same time, one at a time we use. Then you have a mirror which can take light and project it through this, which is called as a condenser. Or there can be even a source of light, a bulb can be put there. These are two adjustments called as force adjustment and fine adjustment knobs, with the help of which the distance between eyepiece and the objective lens and the specimen that you want to observe, which is kept here, the distance can be changed so that a proper image can be seen. The specimen which you want to see can be put here, which is called as a stage. Now, this is a simple understanding of a simple compound light microscope. You see here there are three lenses which are called as objective lens. It's very important to understand that their magnifying capacity is different. One might have 10x magnification, means 10x means it can multiply the image by 10 times. One can have 45x, means 45 times. One can be 100x, means 100 times. Eyepiece again has its own magnification, which is usually around 10 times, called as 10x. If I put here an eyepiece of 10x and the objective of 10x, overall magnification becomes 10 into 10, 100 times. So whatever you put here, you can see an image which is 100 times larger than the actual image. And that is how a microscope is. More you use it in the laboratory, more you will understand about its functioning. So these are some of the pictures which you can see under microscope. You can see this one is little larger than this one. So this is how an onion peel is observed. Is onion of this color, purple color? No. So what we have done, we have put some stains or the dyes which have colored the outer boundaries, and these dots which you see are nothing but the nucleus. These are some of the cells called as human cheek cells from inside your cheeks. From inner walls, they can be scraped out. These are the cheek cells. These are some of the images which are captured not under this compound microscope, but actually these are specialized microscope like electron microscope, which is very high magnification, many thousand times it can increase. So those are bacteria, these are certain viruses, this is a human egg, and these are some more other types of microorganisms. So that was all. Some in-text questions I am discussing here. I am only giving some hints to the questions which are there in your book. Try to make your own answers. Who discovered cells and how? Robert Hooke has discovered using the cord slice. Why the cell is called as the structural and functional unit of life? Because they are the smallest structure which can independently exist. Make a comparison and write down ways in which plant cells are different from animal cells. Actually, in future videos we will discuss about the different cell organelles, where you will learn organelles like cell wall and plastids are present in the plant cells and not in the animal cells. That's why plant cells become more rigid compared to the animal cells. Some more extra questions that we can think of from this part of the chapter, like who observed the living cells for the first time? Recall Leeuwenhoek. Short note on cell theory, those three tenets. What determines the shape and size of the cell? Functions. Which type of cells can change their shape, like amoeba, WBCs, and how do they do it? How does it help them? They do it with the help of pseudopodia. How does it help them? Locomotion, for engulfing food. How are cells visualized under microscope, but only after putting a stain? So these were the different extra questions. There will be two more videos for this particular chapter. In those two videos, we will be discussing about the cell organelles in more detailed understanding. If you have any kind of doubts and you want to talk to me, if you need some written notes on this particular chapter or topic, then you can contact me through WhatsApp in 94353 52781. Thank you, students. See you again in the next video.